Well, yeah, thanks for the invitation. I've been looking forward to this meeting for, well, since I was invited and heard about it, so glad the day has come. Congratulations to the organisers for putting on a great show so far. So I'm going to be talking a little bit about precision prevention. I'll just flag that I'm about to move to the Novo Nordisk Foundation in Denmark as the Scientific Director in Patient Care. So this is my last week in academia, although I will have a part-time appointment at Lund moving forwards. Quick disclosures, like many have had funding from the pharmaceutical industry. I've consulted for the pharmaceutical industry, and I'm an advisory board member and stock owner in Zoe Global Limited. And Zoe Global is the startup that Tim Spector will talk about later, that has run the PREDICT trials, which I'll mention very briefly at the end. So there is a global problem with diabetes. You will all be aware of that. By the middle of the century, 500 million people, men, women and children, will be walking around on this planet with diabetes, the majority of whom will have the more complex form of type 2 diabetes. Some will have type 1 and others will have other variations. But importantly, the vast majority of those 500 million people will not know that they have diabetes. And this is a problem because diabetes causes serious complications that threaten life and impose significant burden on those who have the complications, as well as the healthcare systems that treat them. So we have a huge problem here. Type 2 diabetes, which is what this talk is really about, is a lifestyle disease. It's often associated with obesity. And countries that are migrating from traditional subsistence farming lifestyles to more modern westernized lifestyles, including the BRIC countries, China, India, Brazil and others, will be where the majority of the new cases emerge from over the next couple of decades. We know something about the prevention of diabetes. It generally has an older age of onset. If you're sedentary, you have a family history, you have high blood pressure, if you have a history of gestational diabetes, and most importantly, probably if you're overweight or obese, you are more likely to develop type 2 diabetes. And indeed, the Diabetes Prevention Program, which is one of the most famous studies to use that type of information to enroll high-risk participants, demonstrated eloquently that if you are to intervene intensively with lifestyle intervention, which is shown at the bottom of this cumulative incidence plot, or metformin, which is the frontline therapy for type 2 diabetes, the risk of developing diabetes is substantially reduced. It's 58% risk reduction with lifestyle, 32% with metformin in the DPP at the end of the randomization phase. This is actually from the outcome study, which followed participants now up to 20 years, but these are the 10-year outcome data. Despite the name of the study, which implies that diabetes is prevented, you can see, of course, that many people who were enrolled into the trial and received either lifestyle or metformin still develop diabetes. In fact, at 10 years, it's almost 40% of those populations. And so the other way of looking at this, which is perhaps a little more pessimistic, is to say, what was the delay in diabetes that the interventions incurred? And with lifestyle, it's about 36 months. With metformin, it's 12 months. If you look across other similar trials to DPP using similar interventions, the delay is somewhere in that ballpark. It of course varies a bit, but the point is that it's not years and years and years of delay. It's 3 or 4 years, something like that, and this tracks across the later adult life course. So that's not the same as preventing the disease, and of course prevention is really the ultimate aim. So if we're to move from a really intensive lifestyle intervention, which is wasn't entirely one size fits all, it was tailored to the individual requirements. So for example, the dietary recommendations were tailored to the energy requirements of the individual. And there was, although it was group counselling, there was also one-on-one counselling offered. So it wasn't entirely one size fits all, but the intervention itself was a fairly generic intervention, making the assumption that those risk factors that I showed you on the previous slide will have a similar effect in all individuals. Now, the reason that's perhaps not as effective as one would like is because not everybody responds well to lifestyle intervention. This is now showing data again from the DPP outcome study. It's at 15 years follow-up in this slide. If you look at the bottom of this slide, you'll see the curves for metformin, lifestyle, and placebo are showing the weight trajectories for people who achieved at least 5% weight loss at 12 months post-randomization in each of the 3 arms. Forget about the middle line at the moment, which is the full population, but instead focus on the top line, which are those people who didn't achieve at least 5% weight loss at 12 months. And you can see that with metformin, the fact that they didn't achieve the weight loss really doesn't have any effect on the baseline weight. It tracks over 15 years pretty close, and the same is true for placebo. But with lifestyle intervention, there's a considerable weight gain. These are the y-axis is in kilograms, and so this is a fairly large area under the curve for additional weight accumulation over those 15 or so years follow-up. Now, what we didn't do in this study is to look to see what the consequences of that are, but it wouldn't be too surprising if you found that that additional body weight also conveyed some other health consequences that were unwanted. So we don't know why people didn't respond well to the lifestyle intervention, but we do know that some people don't. And of course, there are lots of other data. Diet Fits is another nice example where people just don't respond well to interventions, be it because they don't adhere well to the interventions, they compensate their behavior, or because of features of their biology that mean that the intervention doesn't work well for them. So within the sorry, my son just walked through the door, got distracted. So a couple of years ago, we set up with the ADA, ESD, NIDDK, JDRF, and others, something called the Precision Medicine in Diabetes Initiative. And this is a figure that's taken from the consensus report published last summer in Diabetes Care and in Diabetologia. And what we did in that paper was really try to put together some framework for what we mean when we talk about precision diabetes medicine. And this is just one of the frameworks that are described in that paper that talks about precision therapeutics, under which precision prevention and precision treatment fall. And in the context of at least how we view precision prevention in diabetes, we're really interested in risk factor avoidance. So these are the things that people are particularly susceptible to that tend to drive blood glucose up. And then lifestyle interventions, the things that people benefit most from. And importantly, not necessarily assuming that those risk factors and interventions will be the same for everybody. I mentioned precision treatment. I won't talk about that now, but one other component to this is monitoring, which is incredibly important, of course, because if you want to know whether or not your interventions are working, and indeed whether treatments are working, you need to monitor. And so the area of sort of precision monitoring, which includes devices and other technologies, is one that is burgeoning. In any case, this is the framework around which the rest of this talk will revolve when we talk about precision prevention. So my own sort of backstory into how I got involved in precision medicine started 20 plus years ago when I was in Cambridge doing my PhD with Nick Wareham and Steve O'Reilly, and we were interested in gene-environment interactions. And of course, back then, pre-GWAS, most of the work that was done was, it turns out, probably to be false positive. So I won't say anything about that. But when GWAS came along, it of course provided new substrate for these types of studies. And we and many, many other people got stuck into really trying to understand whether any of these really verified gene variants interact with environmental and lifestyle factors to modify risk of disease. And so that concept of sort of gene lifestyle interaction is something that, for me at least, was a prelude to the field of precision prevention. And so this is work that we did with Ruth Lewis, Lu Chi, Thomas Kilbelainen, and it was really the sort of the first big G by E gene lifestyle interaction study that was done. Cecilia mentioned FTO earlier, which was the first big hit to come out, or the first hit actually to come out of GWAS, and has stood strong since. And it's also been widely studied in the context of gene-lifestyle interactions. And in epidemiological studies, it does seem to modify the relationship between lifestyle variables and obesity. And what the figure shows here is two subgroups of this large meta-analysis population. Those who reported being physically active at the bottom here in blue, those who reported being physically inactive in orange, the three FTO genotypes, the major homozygous here, the minor homozygous here, and then BMI on the y-axis. And if you squint, look very, very carefully, you'll see that the trajectory across the blue bars is slightly less sharp than it is across the orange bars. And to help you out, I've put the coefficients in here. And you can see that there's a very, very marginal difference in the coefficients, but at this sample size, that's statistically different. Therefore, you have a statistical interaction. Now, that may seem a little bit nuanced, but it was the first replicated example of G by E that have really come out in the context of cardiometabolic diseases. So that's why this, I think, was a particularly important study. Now, of course, what you observe in epidemiology may not be what you observe in intervention studies, because, of course, epidemiological studies are prone to chance bias and confounding often, and they're not interventional. So there are other issues of causal inference, reverse causality, for example. And so what we then did is we went into a bunch of clinical trials, including the DPP, which I mentioned earlier, and looked to see whether this gene variant modified the response to lifestyle interventions in terms of weight change. And depressingly, the answer is no. Anyone who knows how to read a forest plot will see that the summary estimate is overlapping the null, which means there's no significant effect. And I think even in the most optimistic of moods, you'd struggle to see any pattern that might suggest that there's an interaction effect in the trial context. So I think we can conclude that FTO doesn't meaningfully affect weight loss interventions, at least in the context of lifestyle interventions. However, one of the other findings from the large GWAS meta-analyses from GIANT was at the MTIF3 locus. And so we studied this in both the DPP and look ahead. And I should say that we looked at 32 SNPs here. So this is one of 32, but I'll try to convince you this is not false positive in a moment. To make this easy to interpret, I've just stratified out the figures for a second. So I should say that I've already told you a bit about DPP. That's enrolled people who are at high risk with diabetes, but without the disease. Whereas look ahead, which was very, very similar in design, indeed many of the same study centers, investigators involved, but enrolled people who already had diabetes. The G allele at this particular SNP had previously been shown to be robustly associated with heavier BMI. And indeed, when you adjust for baseline weight in a DPP, people who carry the GG genotype are slightly heavier over the course of the 4 years follow-up. But when they're randomized to the lifestyle intervention, you can see, first of all, there's an nadir at 12 months, which is common with lifestyle interventions. People start to regain weight. But the interaction is driven by the G allele now conveying more weight loss and the slower weight loss, slower weight regain, hence the interaction. And that's true also in women and also true in look ahead. So very consistent results between men and women and between the two trials. Now, interestingly, in fact, within weeks, this paper was published from another group in Chicago that had studied an obesity case series and also had looked at this particular SNP and found that the same allele conveys more weight loss retention after surgery. We had another paper from the CHARGE consortium that showed gene diet interactions on BMI. So we're kind of interested in this. And so we've spent a lot of time, a lot of money actually as well, studying the functional basis to this interaction. And what we've done is take an exercise mimetic, which is isoproterenol, and a diet restriction mimetic, which is essentially starvation. And we've exposed, this is for white adipose cells, but also brown adipose cells, which show fundamentally the same thing. Those cells to these mimetics, and then CRISPR edited the cells so that it's a 99% knockdown of MTIF3. And what it shows is essentially that both the basal and the response phenotypes in terms of glycerol release with the exercise mimetic and triglyceride content, or the storage of triglyceride in the adipocyte cells, is affected by knocking down the MTIF3 gene. And the reason for that is because it essentially affects the amount of oxygen consumption. The OCR is the oxygen consumption rate, both at basal levels, also the adenosine triphosphate production, and then also at maximal levels. And so you have this OXPHOS mechanism, which is further illustrated by the effect that knocking down MTIF3 has on genes like PGC1 alpha, which is a master regulator of OXPHOS, this complex that's involved in mitochondrial energy metabolism and many other metabolic processes. So it's a pretty interesting gene, and this is the sort of functional basis to the trial and epi data that I just showed you. Now, of course, genetics gets you so far, and I think I have to say I find it highly unlikely that precision prevention for type 2 diabetes or any other complex trait will be purely predicated on genetics. I think we will have to use other molecular information. This is a review that sort of touches on some of these points that Mark McCarthy and I wrote a few years ago now. But essentially what this figure shows is a mechanism for how environmental signals interact with the genome and at a bunch of different levels, whether it be at the ligand activation, membrane transportation levels, could be that it influences epigenetic variation in the cells, and then of course there are transcription and post-transcriptional regulations. But importantly, these are probably very dynamic types of interactions that are going to be very difficult to model using in vivo epidemiological or clinical trial data. So we certainly have a work cut out, and I suspect that the way forwards will be a combination of different types of science to really try to unpick some of these complex interactions. And we'll need to do that, I suspect, to really understand how to design interventions. Nevertheless, with all of that in mind, it does mean that we can then start to think about other types of molecular biomarkers, and particularly at a protein and metabolomic level, to try to understand how modifiable features of the environment interact with these biological variants to affect health and disease. This is work that we did with Erin Segal and others recently, looking at essentially the factors that influence the serum metabolome, and showed here quite clearly that there are various diet features that influence the metabolome, whether you do that observationally or in the context of intervention studies, in this case with different types of bread. And to some extent, this is mediated by variation in the gut microbiome, and Tim Spector will talk much more about this later. He's also a co-author on this paper. In any case, so this is a kind of proof of concept, if you will, that you can capture information about the background environment, particularly dietary factors, in the serum metabolome. And so what we've also done is then use that to model, this is work with Gary Frost and others in the DIRECT consortium, to model a metabolomic profile for different types of dietary exposure, and then use that to then assess relationships between diet objectively and cardiometabolic features shown over here in people free from diabetes and those with diabetes on the right. So there's lots of ways in which we can use measurable biological variable data in humans, whether it's to objectively model environmental exposures, or as mediators of those environmental effects, as I showed you with genetics a few moments ago. Now, the sort of real step change in understanding, I think, of precision nutrition was really brought forwards by, again, work from Erin Segal at the Weizmann Institute in Israel, when they published this paper in Cell in 2015. They studied 800 young adults intensively for a week. They fed them set meals. They measured all kinds of features of their biology, collected stool samples, and did 16 sRNA on metagenomics. And then they essentially predicted an optimal and a suboptimal diet for a small number of these individuals, about 24 individuals, I think, if I remember correctly. or 26 actually, and then intervened to see whether or not they could outpredict a standard dietary intervention in terms of glycemic response, which they did indeed show was possible. So that really sort of laid the groundwork for other studies that have followed, and one that Tim will talk about, and I will just skim over this quickly. I don't want to steal his thunder, but it's the PREDICT trial, which we published in, I think it was last June. This paper came out, and essentially it's similar to the work that Sigal and others had done, except we expanded the focus to look not only at glycemic response to foods, but also to triglyceride and C-peptide response, but also to extend the number of the range of biomarkers that were assessed and the range of metabolic challenges, the meal challenges that the participants were given. And essentially what it shows is, first of all, of course, there's a great deal of heterogeneity to the standardized meals in terms of glycemic response. People are very different. Over here, it shows that for glucose, there's indeed a fairly large genetic component. This is based on twin data within the PREDICT trial that is important for glycemic response, but for insulin, C-peptide, and triglyceride, the genetic component is small to non-detectable, and so we don't think genetics are likely to be important there. And then using a machine learning approach here, you can see that the important variables are ranked here. Genetics, this is for glycemic response, does indeed rank high when you use SNP-based genotype information. Meal composition is really important, in other words, back of pack information, but then there's a whole bunch of other things that are more contextual. And then over here, you can see that we can predict pretty well an individual's glycemic response to a meal. So I just wanted to end with this message, which this is another figure from the ADA Precision Medicine Initiative Consensus Report. I think many of us are very interested in research and development. We spend a lot of our time doing that, and indeed we spend a lot of money doing that, other people's money doing that. But let's remember that there are a whole bunch of other things that will be important to negotiate successfully to get from discovery through to clinical implementation. And these is just a sort of handful of some of those different barriers that we'll have to negotiate. At the end, I've put in health disparity assessment, which is something that wasn't discussed properly in the first consensus report, but has since become a very important focus for the initiative and the ADA in general. Not least because we recognize that precision medicine has the potential to narrow the gap in health disparities, but it also has there's also a risk that it will widen that gap if technologies and assays and knowledge are not universally accessible. So health disparity assessment, I think, will be an important part on this path to the translation of precision medicine. So just to end up by thanking the many people involved in all of this, including members of my team, funding agencies, and my institution, Lund University Diabetes Centre. Thanks.